Welcome to another screencast where we'll be discussing the regulation of the cell cycle and the checkpoints and what is actually happening at the cellular level, the molecular level here, to regulate the cell cycle. So already before this, you should sort of be able to identify the different checkpoints in the cell cycle. But then by the end of this, you should be able to describe a little bit what's going on there and then explain how these proteins called cyclins and cyclin-dependent kinases are responsible for regulating the cell cycle and how that works. And then if we have time, we'll get to this example here of PDGF. These are platelet-derived growth factors affecting cell division here. Okay, so that's just a specific example of another regulatory mechanism. So if we look at the cell cycle here, the cell cycle is a complex set of stages that's highly regulated with checkpoints which determine the ultimate fate of the cell. So if we look at this diagram here, we have the G1 phase, the S phase, the G2 phase, and then the mitotic phase. So there are several points along the way here. Lots of things are happening in order for that cell to be able to divide and make another copy of itself. So we have these checkpoints as sort of a metaphor here for regulation. Now, the first checkpoint that we want to highlight here is in this G1 checkpoint, and this seems to be the master control one. And if it goes past, if the cell gets past this checkpoint, the cell most likely will divide. It's going to go all the way to completion. If not, if it does not get past that checkpoint, we say then the cell enters sort of a G0 phase. Right? And this cell is not, it's not dead or anything, but it is, say, dormant. It is not, well, dormant's not a great word either. It's just not dividing. Right? So examples of cells that would be in a G0 phase would be things like muscle cells. In humans, things like neurons, nervous system cells that don't really divide, but can be cued to divide, but don't normally divide on their own. Otherwise, most of their cells go past this G1 checkpoint, will then replicate their DNA here in the S phase, replicate and all the other organelles here, and then proceed into mitosis. So this G2 checkpoint here happens after the end of G2 and before, right before mitosis it begins, and then there's a metaphase checkpoint as well. The rest of this screencast is going to be dedicated to looking at this G2 checkpoint here and what's going on there as an example of how the cell cycle is regulated. Now, there were some classic experiments where they took, how do they think about doing this, right? They took cells that they knew were in the S phase and cells that they knew were in G1, and they fused those two cells together. And immediately upon fusing the cytoplasms of those two cells, notice that they did not fuse the nuclei here. When they fused the cytoplasms of those two cells, this cell that was in G1 immediately entered the S phase. So that tells the scientists that something in here, something in the cytoplasm of this cell, when they fused them, was triggering this nucleus here to begin to undergo the S phase, right, and replicate the DNA. So they said that was interesting. Let's take a cell that's in the mitotic phase here, actively undergoing mitosis, grab that cell, fuse it with a cell that's in G1, right, and are there things in the cytoplasm here that will trigger this cell to enter the M phase, right, as it did over here? And lo and behold, sure enough, whatever was in that cytoplasm caused this cell that was in G1 to undergo, to begin to undergo mitosis, right? You had spindle apparatus being formed here. You had microtubules being formed. The nuclear membrane began to disappear. The chromatin condensed into visible chromosomes. Even though the chromosomes did not undergo the S phase, right, or this nucleus did not undergo the S phase, and there was no replication happening there, you could trigger this cell to undergo mitosis just by fusing it with a cell that was actively undergoing mitosis. So then the hunt was on to find out, all right, what's going on in the cytoplasm there? What is happening at the molecular level? What molecules? What's the stuff that's causing those cells to replicate? So at this, or to enter mitosis, at the heart of this, then again, we're looking at what's going on here at the G2 checkpoint. So let me just go to the center and sort of work my way out from this diagram. So here you have G1, S, G2, and M. This is the cell cycle. So this is our time component here. So if we start here and work our way right round, let's see what happens here. We have, at this G2 checkpoint, the scientists isolated a protein that they were calling MPF. Now this is a mitosis promoting factor. No O there. Mitosis promoting factor, also sometimes I've seen it referred to as the maturation promoting factor. But since they were calling it a factor, it tips you off that they didn't really know what they were talking about. I mean, they didn't really know what this thing was, right? I mean, they knew it was a protein, but they didn't know exactly how it was functioning. But they noticed that the concentrations of MPF right here at this point in the cell cycle were extremely high. So then the search was on, all right, what is this MPF thing? And it turns out that MPF is actually two subcomponents. There is a cyclin component, and then there's a CDK component. So this cyclin, right, is another protein. This is a subcomponent of MPF. And then there's the CDK, which is another protein that they were referring to as a cyclin-dependent kinase. Now, a kinase is an enzyme that is involved with ATP and dephosphorylation reactions. Right? And so we'll be phosphorylating, dephosphorylating ATP using ATP. So now what was happening here is that this MPF, it turns out, is an active enzyme. It turns out that CDK right here, this is an enzyme, but this is inactive. As it's shown right here, this shape here is inactive. But when you add cyclin to CDK, when you bind this cyclin right here to the CDK right here, you get this active MPF. So now MPF is an active enzyme. So if you remember back, here's an opportunity to remember what you've forgotten about enzymes. This is not the active site of this enzyme right here, of this CDK. Instead, this is an allosteric site, or so the cyclin could be compared with, say, a cofactor, right? Or a coenzyme. So when cyclin binds to this allosteric site of the CDK, it activates CDK, and then this active CDK is called MPF. Right? So this, you could call this MPF, you could call this active CDK, you could call this inactive CDK. Right? So this active enzyme will then cause something to happen. Right? Well, what does it cause to happen? This active enzyme will cause the signs of mitosis, right? So what are the signs of mitosis? This active enzyme, this active kinase enzyme, sets in motion several different pathways, right? Pathway A, pathway B, pathway C, pathway D, right? And so on and so forth. So one of the things that happens, right? How do you know that mitosis is beginning? Well, nuclear membrane disappears. Nuclear membrane disappears, so this enzyme starts a series of reactions that causes the dismemberment of the nuclear membrane. You know that mitosis is beginning because the chromatin condenses. So another thing that this enzyme does is it sets in motion pathways that lead to the chromatin condensation into visible chromosomes. A third thing that might happen here that you know mitosis is happening is you get the spindle forming, and again, this spindle is microtubules. Right? The spindle from centrosome. Right? So this MPF is an active enzyme. Again, it's causing these things to happen. It's causing right here then this mitosis process to begin. Okay. So the other thing that this MPF does is it also catalyzes the breakdown of cyclin. Right? And so if this enzyme triggers other enzymes that then begin to break down cyclin, right? What you see then is that cyclin is going to be degraded, right? And so cause effect here, no cyclin means no MPF, right? Which means no mitosis, which means all of these things, right? The nuclear membrane will come back, chromosomes will condense or decondense back to chromatin, the spindle goes away, right? And so this degradation of cyclin is a way of turning off mitosis, or to stop mitosis, right? So you get the opposite of mitosis happening there when the cyclin is degraded. So if cyclin is degraded, it can the likelihood of cyclin combining with CDK is extremely low. Now the CDK concentrations tend to remain relatively stable throughout the course of the cell cycle, right? But the cyclin concentrations will change, right? So as the cell cycle goes on, we see here that new cyclin is going to accumulate, right? New cyclin being made by the cell. And as the concentrations of cyclin go up, right, you then have more likelihood, a higher chance of the cyclin combining with CDK. And the timing of this concentration change is just such that everything else is ready to go. You have all the other, you have the S phases happen, you have all the other organelles replicated themselves, etc. Okay? So this is one mechanism that seems to be important in regulating the cell cycle. Okay? So again, when you review this, you want to focus on what MPF is when it's in high concentration, when it's in low concentration. When it's in high concentration, what is it causing to happen over here? Right? What is CDK? What is cyclin? Right? When are their concentrations high? When are they low? And what they are, what they're going to cause to happen there at the G2 checkpoint. So just a couple of review slides. We've seen this slide earlier in the year. So we said that kinases are the things that are doing the phosphorylating, right, and then dephosphorylating. So this step right here would be by some sort of kinase enzyme that would have a specific name that was probably like glutamate or glutamic acid kinase or something like that. But it's going to be taking that phosphate from there, putting it onto here, activating that molecule. Right? So this is how kinase enzymes are working, just to give you a visual. Another visual, talking about the CDK and cyclin concentration. So if this inactive form, let's say that this would be CDK, and then this activator, we could call this cyclin. And then this whole thing with the activator cyclin and the CDK together, this is what we would call MPF. Right? So this is what the activated enzyme would look like. This is what the inactivated enzyme would look like up there. So if you have CDK, cyclin bound to the allosteric site there, and you hold that thing in an active conformation, and that sets off a series of reactions. Okay? Graphically, here's what it looks like. So you can analyze this graph, please, and look at the MPF activity versus the cyclin concentrations. Right? And why does that graph make sense? This is the stuff of a multiple choice question potentially, right? Or part of a free response where you might have to explain why that graph looks the way it does. Double Y graph, again, very similar to this graph up here, but again, looking at protein kinase activity right here in red, and then the percent of the cells dividing. Right? So there seemed to be a correlation there between the two. And that correlation was strong enough to then lead them to say, like, okay, this kinase activity, when it's high, shortly after that, you're going to have lots of cells actively dividing in the mitotic phase. I think we'll stop here. We can talk in class here about the platelet-derived growth factor, the PDGFs, right, and what was going on there in terms of like an external signal, right, to get cells to divide. The MPF was more of an internal signal, right? The stuff that was in the cytoplasm, those molecules that were in the cytoplasm. You can also get cells to divide by having external signals, right? So, chemical signals from other cells, getting them to divide. And again, we can discuss that in class, or you can read that in the text as an example of an external signal. Okay, so what I focused here again was on these checkpoints in the cell cycle. Describe the three of them. Talk about that G1, the G2, the M checkpoint, and then what it means to be in G0. And then explaining how cyclins and cyclin-dependent kinases regulate the cell cycle. We did not really get to this PDGF here, right, as an external signal. Okay, thanks for listening.